Welcome everyone. Good afternoon, and welcome to this Gateway to Translation seminar series episode today. My name is Matthias Silbauer. I'm a clinician scientist here in Cambridge University. I'm based at the biomedical campus, the Stem Cell Institute. I was given permission to just provide a couple of slides summarizing briefly what my lab is interested in, as it's sort of related. So we work a lot with gut organoids. So my clinical background is in pediatric gastroenterology. I've got an interest in inflammatory bowel diseases, and we're using patient-derived gut organoids quite a lot. So for those of you might have not heard it, what we do, we take small tissue samples called biopsies from patients that we investigate for potential gut diseases with an endoscopy. From these biopsies, we can isolate the crypts, put them in a scaffold, and then generate these three-dimensional structures called organoids. And they're extremely versatile. You can perform functional assays, gene editing, molecular profiling. Of course, you can also freeze them. You can generate them from all different gut segments. We routinely sample 3 gut segments. Here you see a video of how each gut segment develops into different, quite unique organoids. So lots of groups have shown that these organoids contain the cells that are in the epithelium in the gut, and they can be differentiated. And as I said, they can be genetically modified. Over the last 10 years or so that we've been working with these models, we've done extensive validation with a focus really on their epigenetic profile, because we believe that epigenetic profiling, especially DNA methylation, provides some sort of cell and donor identity. And we're able to show that organoids retain donor information, including gut segment specificity, age of the donor, the developmental stage, gender. And most recently, we've shown that these organoids also retain information from patients with IBD. As such, they're extremely valuable as a translational tool for drug testing and discovery. Over the last 10 years, we've generated quite a big organoid biobank that contains over 1,000 well-characterized lines. We're now trying to make available to the research community, and which basically is the foundation of a lot of the research that we're doing, including drug testing and drug discovery. And of course, one of the key aspects of these models is the ability to kind of test functional aspects in an appropriate model. And this is where things like organ-on-chip and similar models come in. And at that point, it's a great pleasure to introduce our speaker today. So welcome to Dr. Atefeh Mobasseri. So Dr. Mobasseri is a field application scientist, and she's based at CN Bio. She's got extensive experience and a background in tissue engineering and regenerative medicine. Before she joined CN Bio, she gained her PhD in biomedical and materials from the University of Manchester, and then carried out postdoc positions at the University of Manchester and King's College London, focusing on the investigation and the effect of 3D scaffolds and cellular behavior. And since joining CN Bio, she's been supporting their customers in using CN Bio's range of microfluid systems, and also generating high content human relevant data sets. And today her talk will be on organoids and how we can take them to the next level, state-of-the-art organ-on-chip technology. Atefeh.
Thank you very much for the kind introduction. And also thank you for inviting me to speak today for Gateway to Translation seminar. And as the title of the seminar indicated, we are going to uncover some of the advantage of organ-on-a-chip technology. And also in this time, I'm going to explain about how we can adopt this technology in different applications. So we know that organoids are valuable, and organoids are simplified versions of human organs that we grow them in vitro, and it can recapitulate many aspects of the complex structure of, and also the function of the specific in vivo tissue. And they can self-reorganize in vitro and within the 3D cellular matrix. They provide valuable insight for diverse range of applications, but similar to any other method and models, there are some limitations associated with it. And some of them would be like the differentiation of the cells, the culture longevity, the isolated single organ models, and also limitation of inclusion of complex components such as immune cells. So now the question is that how we can push our in vitro models even more, even further, to have better and improve physiologically relevant in vitro models. So here the organ on a chip, OOC, or macrophysiological system comes into picture. And these technologies are developed by commercial companies such as us, CM Bio, and also there are several academics developing these platforms for specific scientific questions. And they can come in a different design, size, and different technology. The culture environment can be different formats, such as chip, or it can be like a plate. And also some of them include macrofluidic, and the technology to provide the flow would be different. And some companies and some technology incorporated gravity-driven flow. Some provide more tunable and controlled flow. But overall, all of these technology have this one aim, and the aim is that to provide a human relevant model. So usually we can include human cells, and these can be different cell type, primary cells, iPSCs, cell lines, tissue slices, and also it is possible to include 3D macro tissue and create the 3D macro tissue to mimic human tissue structure. Fluidic flow can be incorporated to mimic blood flow and to provide nutrient and oxygen and increase the viability of the cells. And sometimes it is possible to optimize this macroenvironment to really recapitulate the human tissue niche. And the organ on a chip technology or macrophysiological system can be used in academic setup, as I mentioned, and these research can use this technology for developing human physiology for both in healthy models and also disease models. Also, the other application is in drug discovery and development. And for the drug discovery pipeline, the organ on a chip technology can be used at different times. It can be used as early as target discovery, and also it can be used in complement with animal models and in lead optimization. It helps researchers to design the animal studies better and also reduce the number of animals and help us to understand the human relevant and also mechanistic data for the preclinical application. So then one of the main reasons that drug discovery and development process is uptaking these new technologies is that the developing drug is hugely costly, and also it is lengthy. And on top of that, the success rate is very low. So there are a lot of money, time, and energy spent at the beginning of developing of the new compounds to optimize the drug, to screen and target validation, and then so the drug companies need better preclinical tools to be able to optimize their different compounds. On top of that, recently there is a huge progress on developing a new modality drug. So these drugs also is approved by FDA and other regulatory agency more and more. And these drug really needs better and improved human relevant model. And ethically, we all know that we need to reduce the animal usage. There is a change in global and regulatory landscape to transform the way that we use animal model to really, there's a pressure on companies and farmers to reduce the animal and also design their animal models when it is necessary, better and more efficient. So organ-on-a-chip technology comes with in addition to other tools that we have. And all of these tools, in vitro tools, 2D, 3D models, animal models, have their own advantage and limitation. So we need to know when and how we can incorporate these models in our research to take the advantage of its potential and using the technology and these models at the perfect timing for the research progress. So, for example, for the 2D and 3D culture, it is a well-known tool. It is easy to adopt. The human relevancy is moderate, and also the throughput is high. And when we move on to animal model in vivo studies, we know that we can look at more complex process. We can look at the whole body. We can look at the organ crosstalk. However, as we know, the human relevancy is limited. Then organ-on-a-chip technology has better and improved human relevancy in compared to animal model. And also we can look at, to some extent, complex models, and the throughput is medium. So we believe that organ-on-a-chip technology can bridge the gap. It can overcome the limitation of some of the traditional approaches, and also it can give us another method to investigate and include in our clinical toolbox. And it can be used as a new approach methodology tools to give us the better way to for our decision making. I'm going to explain a little bit about Physiomimic's organ-on-a-chip, which is our system, and to give you an idea how this system works. But mainly I'm going to emphasize on the different case studies. So at CM Bio, we offer the organ-on-a-chip technology as a system that it can be used within different labs, and the system provides the flow in a plate. And the plate format can offer different features, and these different features can be used for culturing different tissue models. So we can use this plate, which has the flow to culture liver model, and also different barrier models such as gut and lung model. And also we can connect the two organ models in our dual organ plate to look at the more complex process like gut, liver, or lung liver model. Overall, organ-on-a-chip technology application is versatile. It can be used in disease modeling, and it can be used in drug development for the drug absorption, metabolism, and bioavailability. And also it has been used extensively in area of safety and toxicology. So for our first case study, I'm going to explain the way that we use organoid to develop a disease model of liver disease called NASH, non-alcoholic steatohepatitis. And I can explain in following slides that why we chose organoid, and also how we can design our experiments using organoid, and what are the main readouts, and how we can interpret our data, and what kind of data we can get out of this technology. So NASH is for those of you who are not familiar with NASH, is a severe form of non-alcoholic liver fatty disease, and it is a very complex metabolic disorder in liver, and up to this year, there wasn't any therapeutic for NASH. And this can show the complexity of the disease and the challenges for the pharmas to develop a therapeutic for NASH treatment. So the main reason that preclinical tools are not able to provide a good tool for farmers to develop NASH is that, firstly, as I said, because human NASH is a very complex disease, no single animal model can fully show the spectrum of human NASH progress. And so usually we need several different animal models, designing different animal models to be able to explore NASH in animals. And also for in vitro tools, there is limitation here to fully show the functionality of the liver, and also showing the human NASH phenotype in vitro. So the main requirement here for NASH preclinical in vitro tool is that, first of all, we need improved human relevancy. We need a better way to design the animal studies, and also to have better understanding of mechanistic insight to NASH. Also, it is important when we are having a different in vitro tool, be able to see and explore the expression of a wide range of human translational biomarkers, which are the characteristic of the human NASH. So the way that we design the NASH assay using organoid chip technology is that, firstly, we wanted to be able to culture liver with a flow in our liver on a chip setup. And for this, we included several specific cell type that is important and for studying NASH. So for this assay, we include a primary human hepatocyte, Kupffer cell, and stem cells. Then we switch the media. We switch the media to dietary fat medium. This media helps cells to induce the NASH. So we can, during our experiment, we are able to monitor the NASH disease phenotype, and also understanding about the steatosis, how the lipid accumulate in the cells. We can explore the inflammation and fibrosis, and throughout the experiment, we can look at the liver functionality. So we use our liver plate. We culture the cells. Let me bring up my We culture the cells in the plate, and at each plate we have the 3D scaffold that the cells can grow inside this scaffold and they form the macro tissue. We have the continuous flow around this culture to provide the nutrients and oxygen to the cells, and we have a continuous perfuse. The cells can grow inside this scaffold and form the macro tissue. And we, firstly, for the healthy state, we look at the functionality of the hepatocyte by looking at the expression of different biomarkers and also looking at the expression of albumin. And also we looked at the metabolic activity in our system and see that how we can keep the metabolic activity of the cell hepatocyte for extended period of time when having the flow in the system. So for the NASH, we co-culture hepatocyte the same way that I showed in last slide with the copper cells and stellate cells. We change the media, and then during 10-day culture, we can look at the NASH phenotype and the characteristic of the NASH disease, which firstly is the lipid accumulation. We show that we can see the lipid accumulation and also the fat loading. And also we can look at the expression of inflammatory markers and also fibrosis marker. So we show that, for example, when we quantify the expression of IL-6, TNF-alpha, TIMP-1, procollagen, we see the increase in our NASH group in compared to the hepatocyte group. Also, if we want to look at the progress of the NASH and looking at the advanced NASH, then we're able to add the TGF-beta to promote fibrosis. By this, we can also, again, we can quantify different soluble biomarkers to show that these are the expression of different fibrosis markers when we add the TGF-beta in the system. So in addition to that, we also able to look at the gene expression and the change in the gene expression. We compare this with the human gene atlas. We show that the gene and the transcriptomic profile that we observe in our NASH model is aligned better with the available data for the human NASH. Better, and also it can be comparable with the human gene atlas and when we compare our data with the animal models. So now we know that this NASH model, the model that we have developed, showing the characteristic of NASH, then this is a good time to look at the efficacy of different compounds. We chose 4 different compounds. And these are the compounds that are in the late stage of clinical trials. We use these 4 anti-NASH compounds at 3 test concentration, and the highest concentration is matching the clinical trial dosage. We can really integrate this model, looking at the different expression of biomarkers and correlate these to the action of the mode of action of these compounds to really understand how this compound works in our system, how they can affect on the progress and a different aspect of the NASH. And easily, we can also able to quantify the expression of different biomarkers and also the deposition in the serum. So to summarize, NASH assay is we can create a model using organoid technology that provide us the better understanding of the disease. We can look at the translational biomarkers. We also, because we can get a very high content data out of the system, we have better understanding of mechanistic side. We can compare our results to the animal and human models to understand the human specific response that we can observe in the in vitro NASH assay, and also we can look at the efficacy of different anti-NASH compounds. The second study, case study, is bioavailability. And for those of you who are not familiar with bioavailability, bioavailability describes the proportion of the drug that reaches systematic circulation, and it is an important factor for drug discovery to identify the drug dosage and also the toxicity and safety of the drug. So each compound has a concentration that within this concentration, if we can see the therapeutic window. During an higher concentration, we can expect a side effect, and lower than this, we can expect reduced therapy efficacy. So it is important to understand how these drugs behave in vitro and what are the bioavailability of these compounds. To improve the bioavailability, there are two main factors. We can enhance their oral absorption, and also we can reduce the first pass effect. And the main limitation for exploring bioavailability and predicting human oral bioavailability is firstly the animal models are poor model to provide to understand and show the profile of human bioavailability. This is the study that published in 2014, and this study, they have looked at several compounds and compare their animal bioavailability between different animals to human. And the main thing is that they show the interspecies differences. That is the main cause of the poor prediction of the animal model. And then if we want to look at the in vitro models, usually for bioavailability, KQ2 model is the main in vitro model to be used for investigating bioavailability. However, only KQ2, we cannot see the liver metabolism for the models with only KQ2. Also, KQ2 absence the level of the enzyme and transporters is available in human intestine. So for the requirement here for the preclinical assay is that similar, we need to have the improved human relevancy. Also, a key aspect of exploring bioavailability is to be able to combine oral absorption and also hepatic metabolism. And so the way that we design the oral bioavailability assay is that we culture and develop a primary human intestinal, and then also we culture primary human hepatocyte. Using the dual organ plate, we connect these two tissue models, and we have the media that recirculate around the human intestinal and also hepatocyte. At this point, we are able to start the drug dosing. So we can look at the different regime. We can looking at the different route of administration, for example, mimicking IV or oral dosing. And also we are able to explore different dose and different dosing regime. And then for the readouts, we can do the sensitive LCMS assay that looking at the concentration of the compounds and also the concentration of the metabolite. Throughout the experiment, we can look at the cell health to make sure that we can capture the side effect of the compounds as well. So similar to the NASH assay, we use our plate for culturing hepatocyte, And for the gut liver model that we use for bioavailability, for the liver part, we use a hepatocyte monoculture. And then for this application, firstly, metabolic activity of the liver is very important. So we explore the metabolic activity by looking at the CYP3A4. And also the hepatic clearance is important. So again, this is a published data that we show that the clearance rate of 12 different compounds and how this can correlate to the available human data and the clearance rate of this compound in human. So when we make sure that we can capture the two main parameters for liver related to bioavailability, then we can focus on the gut part. For the gut section, for the gut compartment, we use Replicot. This is the available gut tissue from Atefeh. And we use a Replicot to look at to develop our gut liver model. For the gut only, for this Replicot model, we know that this is the model that use the human jejunum stem cells on the transwell, and these can differentiate it to form a polarized barrier, and then we can see the main cells that we see in human intestine in a physiologically relevant ratio. So also, in addition to that, we compare the Replicot model using the primary gut to KCO2, and also we look at the expression of the specific metabolic enzyme and then compare this to the KCO2 model. Also, in addition to that, we can also measure and quantify the mucus production in Replicot, which is lacking in KCO2 model. So after that, the main challenge here is how we can connect these two tissue models, and mainly because in this model we share the media between gut and liver. So it is important that to have the media that works with both type of cells. So we develop a specialized design co-culture media that we can use for both cell type, and for this we look at for the liver, we look at the albumin production, compare the liver only to gut liver, and also we look at the CYP3A4 metabolism, again compare the liver only to gut liver. We make sure that the liver is healthy. Then we move on to the barrier for the gut. We look at the barrier integrity and also permeability to the supragella. This is the setup that we use for the gut liver, and this is the cross section of one well. Here you can see what we culture the gut tissue in this compartment, which is the transwell compartment. So, and then we culture liver in the liver compartment. We have several pumps that produce the flow. A pump here that produces the flow in the basolateral side. A pump here that produces the perfusion around our liver. And one pump and a macrofluidic that can connect the flow from gut, connect the media from gut to liver. So in this setup, there we have a flexibility to look at different single organ or dual organ models. So we can culture the gut independently, we can culture liver independently, and also we can look at the gut liver. As a proof of concept, when we culture these two models together, we look at the 7-hydroxycoumarine, the 7-HC. And this is the fluorescent compound that undergoes phase 2 metabolism. And we show that how when we add these compounds, when we're looking at measuring the metabolite concentration, we can see difference between the gut only when using the Replicot only, and also gut liver and liver only. So when developing this model, then we start our first case study. And this case study, we use Capril, and this is a prodrug type ACE inhibitor that is converted to its active metabolite and by CYS1 enzyme. And this is an enzyme that is mainly in liver. However, when we look at the expression of these in gut, we see that in KECO2 model, we see the higher expression of the CYS1 in KECO2 model. We know that this drug is designed the way that can be resistant to intestinal hydrolysis. However, if we're using the KECO2 model to explore this prodrug, we can see the highest intestinal hydrolysis and also the high expression of the metabolite. So we know we test this drug in our gut liver model, and we look at the different administration route, IV and oral. For the IV, it's liver only. When we add these, as we expected, that we have the rapid clearance with when we're using this the drug in the liver after 24 hours in the IV dosing. However, when we add these compounds to gut liver with the KECO2 and gut liver replicate, we can see the gut replica gut liver model can correctly show the resistance to intestinal hydrolysis. And again, by measuring the tempo caprylate, which is the metabolite of the tempo capril, we see that we have the highest in our KECO2 as expected. So we see that the human, the KECO2 model is not cannot predict what we can see in human intestine. So to cover and summarize the bioavailability work is that when using this model, we can see limited interspecies differences as we are using primary cells for both primary human hepatocyte and gut compartment. We are able to combine these two together in the same experiment, so we can looking at the combination of intestinal absorption and hepatic metabolism, and also looking at the different concentration of the drug. We can look at how this drug behave and how this drug interact with both gut and liver. And also on top of that, for some compounds like prodrug, this would be improved way to profile bioavailability in vitro. And let me check the timing. Okay, I still have time for the last case study, which is safety and toxicology. And for this study, we are mainly focusing on the drug-induced liver injury. As I mentioned at the beginning, the failure of the drug in a clinical trial is high, and 80% of that is due to hepatotoxicity. And DILI is the cause of 50% of acute liver failure, and also with the mortality rate of 10%. It is a serious condition. This is a serious challenge, and the same way thinking that there are challenges around investigating DILI, and we need a model to be able to see the expression of the target in target organ, also expression of targets in other organs, and also looking at the metabolic activity, and also if we can include immune system component. For the physiomimics DILI assay, we use the liver cells, and for the liver cell, we combine and co-culture primary human hepatocyte and culprit cells. And then for 4 days, we culture them together. We measure the health of this culture by looking at the functionality, and also looking at the metabolic activity. Usually we look at the LDH, albumin, urea, and CYP. When we make sure that the cells are healthy, happy, then we can introduce the drug in the system. When we introduce the drug, the beauty of the system is that we can do the repeated dosing. We have the flexibility of doing the repeated dosing. We have the flexibility of sampling, repeated sampling during the experiment. So we can add the drug and then start looking at how this drug affects on the hepatocyte. And then by including different endpoints and also looking at the different suitable biomarkers, we can understand the hepatic signature of different compounds in our culture. One classical example of testing the system and validating this model is using troclodoxime. And troclodoxime was in the market and approved by FDA in the market, and it was used for a couple of years, but it was withdrawn because of its delay concern. And so we chose these compounds. We added these compounds in our model, and we see that we can, in different ways, we can look at the toxicity effect. We can look at the metabolic effect of this compound on the metabolic activity, on the albumin production, and also on top of that, we can look at the clinical biomarkers. This is a very important step in vitro tools, that if we are able to use the same way that we look at these compounds in clinical, we can have the better understanding of the compounds and better translation from the in vitro to in vivo. So we can also include ALT, AST, and we see that how these compounds show and the toxicity of this compound in vitro. And this is not only for the troclodoxime. We measured and added the different compounds. There is a IQ consortium, Innovation and Quality Consortium. This is this is between several big pharmas that they published a way to test the delay sensitivity test and different compounds to be used for this sensitivity test. We chose several of these compounds. We added in this in our model. We look at several different ways to look at the toxicity, and we show that using this organoid technology, we can pick up the difference between the toxicity of different compounds, even with the low delay concern. And combining toxicology with pharmacology, as I mentioned, because we are able to do the repeated sampling, and relatively we are working with large amount of media during the experiment, we are able to do the multiple sampling. We are able to correlate the different assay during one experiment. So for example, one example that we showed here is acetaminophen, that we know the metabolite formation profile, and we can see the human specific metabolite, and then we can detect this in our model. We can profile the metabolite formation in relation to the time and the concentration, and also we can correlate this with the cell health assay. So really we can correlate the metabolite formation with the toxicity during the experiment, so at different times. So to summarize the daily work, we are able to include different translatable, clinically relevant endpoints, and because we get a high content data from this technology, we can have and explore the mechanistic insight. Also, human-specific response is observed in our culture, and all of this can help the researcher to refine the preclinical experimental design. So to conclude the seminar, I hope today I can uncover some of the advantage of organoid chip technology in terms of giving us more confidence, in terms of giving us the more human relevant investigative tool, and reducing basically our costs, and it would be a very cost-effective model. And to conclude, this is the complementary tool. This is the complementary tool that that has the potential to transform the way that we generate our human relevant preclinical data. And thank you very much for your attention. Happy to take any questions. Thanks.